Well, hi everybody. Welcome to the week 12 review video. Although this is the second to last video that you all will be watching, it's actually the last video I'm recording for the entire semester. I explained this in the challenge question recap video as well, but I always record the challenge question recap videos sometime on Saturday because I don't really need your feedback. I know what the challenge questions are. It's easy for me to go through them and recap them for you. And then I wait until later in the day on Sunday to record the review video so that I have a chance to get as much feedback from you as possible. So although you will be watching the challenge question recap video after this one, I'm recording this video as the last video of the semester. So bittersweet moment. I kind of get a little bit gushy in the challenge question recap video as well, but it's been quite a semester for all of us. Really, I think probably the roughest and most challenging semester that any of us, including me in 15 years of experience being a college professor, has ever experienced. So we deserve a collective pat on the back. I think we had a wonderful course. We had wonderful learning. We had robust discussions, deep learning. I think we proved that this format of teaching and learning can work in almost any modality. I know it was harder. I know it was a lot more to be asked of you. Required a lot more time management and personal responsibility, and I know you had so many other things going on. My heart goes out to every single one of you, but we did the absolute best we could in challenging circumstances, and so kudos to all of us. And that's enough of that, though. So what we're going to do in this video is basically recap all of photosynthesis, try to do what we've done for glycolysis and the citric acid cycle, all of glucose metabolism, just kind of go through the entire process, strip away all of the non-chronological side stories and side points, and just get to the bare bones of photosynthesis, the light and dark reactions. Now that's going to be quite a bit of material, even stripped down, because as you've seen already, so much goes on, but at least it will bring this story of photosynthesis into a cohesive whole for you if you're missing that. And then at the end of that, we'll go over the C4 pathway for tropical plants and how tropical plants get carbon dioxide deep into their tissues. That's separate from photosynthesis. It's really just capturing and regenerating carbon dioxide, but we'll cover that at the end of the video. All right, so let's dive in. So what you've all come into this with, if nothing more, I know some of you have had botany and you conveyed that in your feedback, and that's awesome, but what we've all known since maybe elementary school is that plants really need three things to survive. They need, they need water, carbon dioxide, and sunlight. If you give a plant these three things, it'll survive. Water, carbon dioxide, and sunlight. And what we see as we go through the deep biochemistry of photosynthesis is where each of those elements plays a role in allowing photosynthesis to be possible. So let's start off with the light reactions. And the light reactions, of course, is starting off with sunlight. So sunlight hits the plant. It penetrates into the cells, and it ultimately lands, the sunlight lands, on the thylakoid membrane. These are the membranes of the thylakoid discs. These discs stack to form grana, and this is all in the chloroplast of the plant cell. So on that thylakoid membrane, embedded in the membrane, is chlorophyll. Chlorophyll, the green compound that captures sunlight. So this sunlight lands on chlorophyll, and more specifically, it lands on antenna chlorophyll. And eventually, that sunlight is going, eventually that energy, that captured charged energy from sunlight is going to move from these antenna chlorophyll to a reaction center. A reaction center is just a portion or a region on the thylakoid membrane. So if this is the thylakoid disc in profile, let's imagine the thylakoid disc. Now we're looking down on top of it. You have antenna chlorophyll, antenna chlorophyll, antenna chlorophyll making up the vast majority. And then there's a little region here that's a reaction center. So you have relatively few reaction centers as compared to antenna chlorophyll. And as the sunlight lands on that, and that sunlight is captured as electron energy in the antenna chlorophyll, that energy is rerouted and pooled until it lands at a reaction center. So what is a reaction center? Again, a reaction center is just a region on the thylakoid membrane, but it's a region that contains two photosystems, photosystem 2 and photosystem 1. So these two photosystems and the electron transport chain between them makes up the reaction center. So essentially what we're really saying here is that sunlight lands on the thylakoid membrane, and most of it is captured by antenna chlorophyll. And then that sunlight energy moves to a reaction center where it encounters photosystem 2. So we just got sunlight energy to photosystem 2 on the thylakoid membrane. So that sunlight energy is then used by photosystem 2 to break water. To break water. So there's the water requirement. This is why we have to water our plants. That's the water, right there in the first step of photosynthesis. And what the plant doesn't need is the oxygen. So the oxygen is released as molecular oxygen. This is the waste product of this first step of photosynthesis. This is the oxygen that we breathe. What the cell was really after, what the plant was really after, were the electrons. So we harvest 4 electrons, and 4 protons solely because where electrons go, protons tend to follow in biological systems. So we take water, we crack that water open, we harvest the protons from the water, we harvest the electrons from the water, and we release the oxygen as waste. And what photosystem 2 then does to this set of electrons is it charges them. It adds energy to them. In fact, that charging, that addition of energy, happened as a byproduct of breaking those electrons off of water. To get those electrons to leave water, they had to have energy added to them. So these electrons are now more energetic than they were when they were part of water, because to remove them from water, energy had to be added to these electrons. So they have a higher electron potential. Okay, so to recap, sunlight energy was captured from antenna chlorophyll. It made its way to photosystem 2 at a reaction center. Photosystem 2 took water, the water that you poured into the pot to keep this plant watered. It took water, and it stripped that water of electrons by putting energy into those electrons, and the protons left as well. What was left over, molecular oxygen, is released as waste. So now we have the electrons and the protons. Now we can charge those electrons, we just did, to get them off of water, but we can't charge them enough. So now those electrons are shuttled over to photosystem 1. And photosystem 1 is going to accept those 4 electrons. Now some energy was lost here in that shuttling. So now the energy level of the electrons is probably somewhere here. In order to get those redox reactions to occur, in order to change those electrons' hands from photosystem 2 to some electron carriers to photosystem 1, we had to lose some energy along the way. But those electrons arrive at photosystem 1 more charged than they were when they were part of water. And photosystem 1 takes sunlight energy and charges those electrons. Photosystem 1 is also part of the reaction center, so it's getting that same sunlight energy that arrived from the antenna chlorophyll. And now I know the top of that arrow is further than you can see, so right here is your top. So now we have fully charged these electrons. We've gone from a ground energy state when they were part of water all the way to this high energy state. We charged them partially in photosystem 2, lost some of that energy when we transferred them to photosystem 1. Photosystem 1 then charged them some more and made them high power electrons. At this point, those electrons, as well as the protons that we harvested, can be added to NADPH. NADPH is an electron carrier for high-powered electrons, and now NADPH is reduced. It's carrying the electrons and protons for us. This is one end product of the light reactions. We're going to use that NADPH in the Calvin cycle. Additionally, photosystem 1 also takes the energy from the sun, and it uses that energy to pump protons across the cytoplasmic gradient. So we have a region where proton concentration is low and a region where proton concentration is high. And there is an ATP synthase-like enzyme in plants, and those protons are going to flow through that ATP synthase enzyme, and they're going to cause that ATP synthase enzyme to spin, and that's going to make ATP for us. That ATP is being made powered by sunlight energy. That ATP is being made because a proton gradient is being formed using energy from the sun by photosystem 1, and then that proton gradient energy is being released as it flows through ATP synthase. So ATP is another byproduct of the light reaction. So what are we getting out of the light reactions? We're getting NADPH in its reduced form, carrying high-powered electrons. we're getting ATP. We're going to use some of this ATP in the Calvin cycle, but the rest of this ATP the plant is actually living on. And we, now I'm being very human-centric here, very metazoan-centric, and we get oxygen. The oxygen was waste for the plant, but we need it, we use it. So the oxygen comes out there. So that's the light reactions broken down, as stripped down and as simplistic as I can get them. To recap the whole process, light energy is gathered by antenna chlorophyll on the thylakoid membrane, and then shuttled and pooled to a reaction center on the thylakoid membrane. What's in a reaction center? Photosystem 2, photosystem 1, and this ATP synthase-like enzyme. Photosystem 2 uses that sunlight energy to charge the electrons of water. And those electrons become so charged that they sever themselves from water, and the protons leave as well. So electrons and protons are harvested from water, and those electrons are high energy now because of the energy from the sun that they've received. The rest of water, molecular oxygen, is released by the plant as waste. Those charged electrons that we just stripped off of water are passed from photosystem 2 to photosystem 1 in negative delta G redox reactions, so those electrons lose some of their energy. But they arrive at photosystem 1 more charged than they were when they were part of water, so we lose some of the energy, but not all of it. Photosystem 1 then takes additional sunlight energy from antenna chlorophyll and uses that energy to supercharge those electrons even more. Now these high-power electrons are used to reduce NADP+, giving us NADPH. Photosystem 1 is also using excess sunlight energy to pump protons. This proton gradient is used to power an ATP synthase-like enzyme, loose tight open, loose tight open, loose tight open, and the cell is making ATP as well. The plant needs this ATP to survive in general, but some of this ATP is going to be used in the Calvin cycle. This NADPH is going to be used in this Calvin cycle, and this is going to be breathed in by us to serve as a trash can for spent electrons at the end of the ETC. But the plant doesn't care about this. So, again, that's the light reactions. Now let's move straight into the dark reactions. Dark reactions do not occur only at night. The dark reactions occur all the time. They just don't need light. So keep that ATP and that NADPH plus H in the back of your head. We're going to need that in a little bit. But now, for the dark reactions, we're going to use that last thing that all plants need. Let me start with this list again, because sometimes we get so much into the weeds that we forget the big picture, right? Well, we just saw where water is used. Water is the source of electrons. And we just saw where sunlight is used. Sunlight charges those electrons and makes ATP. The last thing that plants need that we haven't covered is carbon dioxide, and we're going to see that in just a second. So in the chloroplasts, there is a molecule called ribulose 1,5-bisphosphate. Ribulose is a 5-carbon sugar, very related to ribose, which you know from DNA. It's a similar sugar, ribulose, and we know what 1,5-bisphosphate means at this point. There's a phosphate on the 1 carbon of ribulose, there's a second phosphate on the 5 carbon of ribulose, so it's a bisphosphate. So ribulose 1,5-bisphosphate is going to start off our Calvin cycle. And for the Calvin cycle, we're going to be using 6 of these simultaneously. Pretty incredible. Are you getting blurry? Hold on, let me autofocus. That just made it worse. There we go. And again, these are 5-carbon molecules. Each of these is a 5-carbon molecule. So if we've got 6 5-carbon molecules, we're playing with 30 carbons right now. Let's track the carbons. The very first step of the Calvin cycle is to accept 6 carbon dioxides, 6 CO2s. Let me move that down. 6 CO2s. That's the carbon dioxide that plants need to survive. Now we've just accounted for all 3 things that plants need. Water and sunlight and light reactions, and the carbon dioxide here in the dark reactions. When we add a carbon dioxide to each of these ribulose 1,5-bisphosphates, so 6 ribulose 1,5-bisphosphates accept 6 carbon dioxides, 1 carbon dioxide per ribulose 1,5-bisphosphate. When that happens, we make 6 very unstable intermediates. We're not even going to name them, because they're so unstable. They're so unstable that they immediately collapse into 12. Each one of them splits in half. So if 6 molecules split in half, 6 things each cut in half is now 12 things. We get 12 3-phosphoglycerates. We'll recap that in just a second. But I want to keep in mind that if we had 30 carbons in these ribulose 1,5-bisphosphates and we added 6 carbons in 6 carbon dioxides, then we had 36 carbons here. And 3-phosphoglycerate, hopefully you remember from glycolysis, is a 3-carbon molecule. So if we have 12 3-carbon molecules, we have 36 carbons here. So we haven't lost any carbons. So let's recap that first. 6 ribulose 1,5-bisphosphates. Let's represent them. Here they are. 6 ribulose 1,5-bisphosphates. They're each carrying 5 carbons. Each one of them condenses with a carbon dioxide. There are 6 carbon dioxides, each condensing with those ribulose 1,5-bisphosphates, and each of those carbon dioxides is carrying 1 carbon. So now we have 6 6-carbon molecules, right? This is a 6-carbon molecule, this is a 6-carbon molecule, these are the intermediates, these very unstable intermediates. But each one of these now spontaneously splits in half and becomes 2 molecules of 3-PG. So instead of this 6-carbon intermediate, we get 2 3-PGs. Instead of this carbon intermediate, we get 2 3-PGs. And instead of this one, we get 2 3-PGs. And you get the point. So that would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 3-phosphoglycerates. That's what happens in the beginning of the Calvin cycle. And this is catalyzed by our enzyme, Rubisco. Rubisco short for its full name, but we only need to know it by Rubisco. Rubisco is responsible for this first step of the Calvin cycle. So each of these 12 3-phosphoglycerates is going to be phosphorylated and become 1,3-bisphosphoglycerate. Not a confusing molecule for us because we've seen that before. That means we needed the addition of a phosphate to go from 3-phosphoglycerate to 1,3-bisphosphoglycerate. We needed a phosphate, and that phosphate came from ATP. This is ATP made from the light reactions from photosystem 1. And how many 1,3-bisphosphoglycerates do we have? 12 of them. We have 12 3-PGs, we have 12 1,3-BPGs, and we have still 36 carbons total. Now, all 12 of these 1,3-BPGs are going to become GAP, glyceraldehyde 3-phosphate, another old friend from glycolysis. Remember we had 1 DHAP and 1 GAP, and we isomerized the DHAP to GAP, and then from that point on we had glycolysis in parallel. Well, this is glyceraldehyde 3-phosphate, same molecule. In order to go from 1,3-BPG to glyceraldehyde 3-phosphate, we actually need a redox reaction. This is the reduced form of 1,3-BPG. And that redox reaction, that's going to be catalyzed here. The cofactor for that is going to be, well, there it is, NADPH. The NADPH we made in the light reactions was necessary to make these 12 GAPs. Okay, so 30 carbons total here. We added 6 more, 36 carbons in these intermediates, 36 carbons in these 12 3-PGs, 36 carbons in these 1,3-BPGs, 36 carbon in these 12 GAPs. We haven't lost any carbons. Now we diverge. We're going to take 2 of these GAPs, and with them we're going to do essentially gluconeogenesis. We're going to take one of those GAPs and leave it as a GAP. We're going to take the other GAP and isomerize it to DHAP. We're going to take the GAP and the DHAP together and use it to make fructose 1,6-bisphosphate. We're going to dephosphorylate that to fructose 6-phosphate. We're going to isomerize that to glucose 6-phosphate, and then from that we're going to make glucose. There it is. The whole point of photosynthesis, making glucose from nothing but carbon dioxide, water, and sunlight. The meat of the Calvin cycle is in the regeneration of the ribulose 1,5-bisphosphates. We're going to spend a decent amount of time on that in a second, but I'd like to track this one pathway because essentially this is the point of photosynthesis. Although it's the simpler portion of the Calvin cycle, Sometimes instructors go over this too briefly because it is relatively simple, but it's the point of photosynthesis, so let's kind of summarize it again. 6 ribulose 1,5-bisphosphates condense with 6 carbon dioxides to give us 6 6-carbon unstable intermediates, 36 carbons total. Those 6 intermediates break down into 12 3-phosphoglycerates. We phosphorylate those 12 3-PGs to make 12 1,3-bisphosphoglycerates, 12 1,3-BPGs. And then we're going to reduce that. In a redox reaction, we're going to convert those 12 1,3-BPGs into 12 GAPs, 12 glyceraldehyde 3-phosphates. In order to go from this step to this step, we used ATP from the light reactions. In order to go from this step to this step, we used NADPH from the light reactions. Keep in mind, this electron carrier is carrying those supercharged electrons that were stripped from water. And this was made with the proton gradient created by photosystem 1. Of these 12 3-phosphoglycerates, 2 of them are taken. One is left as glyceraldehyde 3-phosphate. The other is isomerized into DHAP. Then those 2 molecules are brought together to make fructose 1,6-bisphosphate. That's dephosphorylated to make fructose 6-phosphate. That's isomerized into glucose 6-phosphate, and that's dephosphorylated into glucose. This is nothing more than gluconeogenesis. It's just occurring in a chloroplast. You've learned this. But the end result is glucose. Glucose made from carbon dioxide and energy from the sun. Now that's the catch, right? The carbons of this glucose came from carbon dioxide. These 6 carbons are the 6 carbons in this 6-carbon glucose. So carbon dioxide was converted into sugar. But to do that, you needed a lot of energy. This energy came directly from the sun. Sunlight energy was used to make a proton gradient that allowed this ATP to be made. And the electrons being carried by this mobile electron carrier are charged, superpowered electrons because they contain sunlight energy. Pretty awesome. But glucose is the end result. Now that's all fine and good, but what we're left with at the end of that, at this step, is the other 10 glyceraldehyde 3-phosphates, right? We made 12 glyceraldehyde 3-phosphates. We used 2 of them to make glucose. We have 10 left. And this can't sustain itself. This can't continue unless it's a cycle. We started with 6 ribulose 1,5-bisphosphates. We need to end with 6 ribulose 1,5-bisphosphates. And right now, we're just holding on to 10 gaps. So what do we do? Well, we have to regenerate ribulose 1,5-bisphosphate. So the first portion of that is called preparation. So this is the preparation step. Preparation. And in the preparation step, some of those gaps, now we're going to stray away from the numbers a little bit. Let's just keep in mind that each of these glyceraldehyde 3-phosphates is made up of 3 carbons each. So we are playing with 30 carbons here. And it's worth making the point that what we're trying to get to is 30 carbons, and what we have now is 30 carbons. So we really just have to, this is oversimplification, but have to do like a mega isomerization to turn these 10 gaps into 6 ribulose 1,5-bisphosphates. We've got 30 carbons here, 30 carbons here. We're just trying to get from A to B. The remaining 6 carbons that we had here and here and here and here, those 6 carbons are here in 6-carbon glucose. So first step, preparation. Some of those gaps are isomerized into DHAP. Others are left as gap. That's the preparation step. And then those DHAPs and those gaps are brought together to make fructose 1,6-bisphosphate. Well, nothing new to remember there. We did that over here. That's gluconeogenesis. And then we take that fructose 1,6-bisphosphate and we dephosphorylate it into fructose 6-phosphate. Looking pretty good. But we don't do that to all of these. We're only doing that to a subset. So then we take some of that fructose 6-phosphate. We take some of the gaps that we left alone. And we take some of the DHAPs that we left alone. So again, keep in mind, we didn't isomerize all of these gaps into DHAP. We only did it for some. And we didn't link all of these gaps and DHAPs into fructose 6-phosphate. We only did that for some. So we reserved some gaps and some DHAPs, and now we're playing with those along with fructose 6-phosphate. And we take these three components, GAP, DHAP, and fructose 6-phosphate, and we reshuffle them. So this is the reshuffling step. Reshuffle. And when we reshuffle those, we make a lot of strange molecules that we haven't encountered in this course yet, and we're not even going to name them. I'll just put down as placeholders their abbreviations. But we have E4-phosphate, we have an intermediate SBP. SBP is used to make S7-phosphate. We just have these kind of weirdo intermediates. These are the end products of this reshuffling. You can think of it along the lines of we took GAP, DHAP, and fructose 6-phosphate and broke them apart into their substituent groups and reassembled them into these molecules. Then S7-phosphate is isomerized. So this is our isomerization step. Into two separate molecules. We make some xylulose, 5-phosphate, and we make some ribose 5-phosphate. In fact, we make 2 ribose 5-phosphates and 4 xylulose 5-phosphates. And then each of these is going to be isomerized into ribulose 5-phosphate. Now, ribulose 5-phosphate is a 5-carbon sugar. So are these. These are 5-carbon sugars as well. So if we take 4 xylulose 5-phosphates and we isomerize it into 4 ribulose 5-phosphates, if we take 2 ribose 5-phosphates and we isomerize those into 2 ribulose 5-phosphates, then this gives us a total of 6 ribulose 5-phosphates. Each of them is a 5-carbon sugar. So there's our 30 carbons still. And then the last step is a phosphorylation step. It requires ATP for that phosphorylation, and when we phosphorylate ribulose 5-phosphate, you're probably not surprised to learn that we will make ribulose 1,5-bisphosphate. We had 6 ribulose 5-phosphates. We phosphorylated all of them to give us 6 ribulose 1,5-bisphosphates. Those are 5 carbons each. And those are the 6 ribulose 1,5-bisphosphates, let me use a different color, that are then ready to condense with 6 more carbon dioxides and do the process again. We've come full circle. We've reestablished the Calvin cycle. All right, so let's kind of strip this away. We talked about this and already recapped it. Let's strip that away and focus on the regeneration portion of the Calvin cycle. So we start with 6 ribulose 1,5-bisphosphates, 5 carbons each, 30 carbons total. Each one condenses with a carbon dioxide, so 6 carbon dioxides come in, giving us 36 carbons total, 6 intermediates. Those 6 intermediates, they split in half spontaneously, giving us 12 3-carbon 3-phosphoglycerates, 36 carbons. We phosphorylate all 12 of them to give us 12 1,3-BPGs. And then we reduce those 12 1,3-BPGs to give us 12 GAPs, 12 glyceraldehyde 3-phosphates. We still got 36 carbons here. We take 6 of those carbons, 2 of those GAPs, and we use it to do a glucogenesis-like process and make glucose. But the remaining 10 GAPs, so now they're 3 carbons each, that's 30 carbons, go through this regeneration scheme. Some of those GAPs remain GAP. Some of those GAPs are isomerized to DHAP. Some of those GAPs and DHAPs are brought together to make fructose 1,6-bisphosphate. Others remain GAP or DHAP. So right now, we've got some GAP, we've got some DHAP, we've got some fructose 1,6-bisphosphate. All of that fructose 1,6-bisphosphate is dephosphorylated to make fructose 6-phosphate. Now, the fructose 6-phosphate we made, the GAP that we have, and the DHAP that we have are brought together in a reshuffling reaction to make these intermediates, these 3 intermediates, which are complicated. We're not going to name them. These are part of the pentose phosphate pathway. No point in naming them. They're just intermediates. But ultimately, this intermediate is going to be isomerized into 4 xylulose 5-phosphates and 2 ribose 5-phosphates. Xylulose 5-phosphate is a 5-carbon sugar, so this is 20 carbons here. And ribose 5-phosphate is a 5-carbon sugar, so this is 10 phosphates here. 20 carbons, excuse me. 20 carbons here and 10 carbons here, 30 carbons total. The same 30 carbons we had here. Those 4 xylulose 5-phosphates are going to be isomerized into 4 ribulose 5-phosphates. These 2 ribose 5-phosphates are also going to be isomerized into 2 ribulose 5-phosphates. 4 ribulose 5-phosphates from here, 2 ribulose 5-phosphates from here, gives us a grand total of 6 ribulose 5-phosphates, each of them with 5 carbons, still 30 carbons. All 6 of those ribulose 5-phosphates are going to be phosphorylated using ATP from photosystem 1 to give us ribulose 1,5-bisphosphates. There's 6 of these, we make 6 of these, and that's the 6 that we started with. We've regenerated the Calvin cycle, and we're right back to the beginning. And that's photosynthesis. So, the point of this is to make glucose, is to do this 2-gap kind of side reaction that ultimately makes glucose for us. That's the point of all this. Everything we're talking about here, that's just regeneration so that we can do it again and do it again and do it again and do it again. The glucose that we make, a lot of it's going to be used to make the cell walls of those plant cells, the cellulose fibers. A good amount of it will be used to make starch, amylose and amylopectin, which the plant can then feed off of when times get hard. So, that is the Calvin cycle. That's the essence of the dark reaction. And now we see why plants need water, because it's a source of electrons. Why plants need sunlight, because it's that sunlight energy that gets put into those electrons and also makes a proton gradient to create ATP. And why they need carbon dioxide, because it's the carbons of those carbon dioxides that will ultimately become glucose and allow the plant to regenerate ribulose 1,5-bisphosphate. So the last thing I wanted to review for you was the tropical plant C4 pathway stuff. Again, this isn't something that most plants do. This is just for tropical plants, plants that exist in a very, very warm climate. But a number of you did say in your feedback to me in Edpuzzle that this was something you found confusing, so we want to review it. The whole point of this C4 pathway is to get carbon dioxide deep into the plant. So we're going to start with carbon dioxide in the environment, and we're going to end with carbon dioxide deeper in the plant. That's all we're really doing. We're shuttling carbon dioxide. So I always like to start off with the grand goal. This carbon dioxide is going to be taken from the environment in through the surface plant cells, and it's going to be condensed with phosphoenol pyruvate, old friend of ours. And when you condense carbon dioxide and phosphoenol pyruvate, you make oxaloacetate, another old friend of ours. That oxaloacetate is going to be reduced into malate. This is almost like the last step of the citric acid cycle in reverse. Malate becomes oxaloacetate and we steal electrons, and in oxidation, here oxaloacetate is being reduced to malate. We are reducing oxaloacetate into malate. And probably not a surprise to you, that's a high power reduction, so we're taking electrons from NADPH, the electrons that we've charged from the sun. That malate is then going to be shuttled deeper into the plant. And that malate is carrying carbon dioxide. It's our carbon dioxide shuttle. Once it gets into those deeper plant tissues, that malate is going to be decarboxylated. Great. There it is. Carbon dioxide is released deeper into the plant. Now it can't escape in that hot weather, and the plant can use it for the Calvin cycle. What's left after that decarboxylation, however, is pyruvate. Pyruvate. That pyruvate is going to shuttle back into the surface cells. So I'll say surface of the plant, I guess. Back to the cells that captured the carbon dioxide in the first place. And there, in the surface cells, it's going to be phosphorylated with ATP, ATP made on the light reactions, and phosphorylated pyruvate is phosphoenol pyruvate. And we've kind of gotten this cycle to regenerate. And that's really it. So carbon dioxide comes from the environment and condenses with PEP. That gives us oxaloacetate. Oxaloacetate is reduced into malate using NADPH as the electron donor. That malate is shuttled deeper into the plant tissues. In the plant tissues, that malate is decarboxylated. This carbon dioxide then goes into the Calvin cycle. It's going to be used to condense with ribulose 1,5-bisphosphate, and down the Calvin cycle we go. That decarboxylation results in pyruvate being deep in the plant tissues. That pyruvate is shuttled back to the surface cells of the plant, where it's phosphorylated back into PEP, and the cycle can continue. And that's it for the C4 pathway. And that's it for photosynthesis, and I guess that's it for me recording videos. Man, pretty crazy. So hopefully photosynthesis is clearer to you after this. It is my genuine hope that as complex as it is, and I have a full appreciation, especially for the Calvin cycle, of its complexity, hopefully said in this kind of 30-minute span as one solid story, it has more context and a little bit more clarity. But you're all pros at this point. You know exactly what to do if that's not the case. Go ahead and let me know in the dialog box that's going to pop up right now.